Hello, everybody. In this lecture, we're going to talk about horizontal gene transfer. Let's get right into it. By the end of this module, you should be able to compare horizontal and vertical gene transfer, explain plasmids, why they're important in nature and lab, describe conjugation, explain how it contributes to genetic diversity, discuss the features of a cell that can conduct conjugation, define recombination, explain how this process leads to production of HFR strains, describe Griffith's experiments, compare and contrast specialized and generalized transduction, describe the two main categories of transposons. All right. So when we think of gene transfer, generally it's vertical gene transfer. So that's parents to children, or in microbes, that's progenitor cell to daughter cells. And so that's what we see in the left-hand side of this image. However, you can have horizontal gene transfer, which is passing genetic information between cells that's independent of cell division. Comes in a couple formats: transformation, transduction, conjugation, and then this other form, which is facilitated by something called transposons. And so here we go, our four types: conjugation, transformation, transduction, and transposons. So plasmids are involved in horizontal gene transfer, not in all types, but they can be. And cells can use plasmids to have genetic information that is outside of their normal chromosome. One type of plasmid that is important both clinically and in the laboratory is the R plasmid. And the R plasmid is any plasmid that contains tetracycline resistance. So we see that contains any sort of resistance. In this, we see that there are multiple resistances included on this plasmid. So we've got sulfonamide, streptomycin, chloramphenicol, fusidic acid, tetracycline, and mercury. That's what the Hg is. So this is in line with what we've been talking about in lab a little bit, where you get metal resistance conferred at the same time as you get some sort of antibiotic resistance. So in this case, the metal resistance that we have conferred is going to be the mercury, and then the antibiotic resistance, well, there are couples: tetracycline, chloramphenicol, streptomycin, and sulfonamide. I'm not sure if fusidic acid is a antibiotic or not. And so clinically, obviously, this means that organisms that have resistance to these antibiotics prevent them from being treated by those antibiotics. In the lab, this is useful because if we give a cell an antibiotic for, say, tetracycline, or a resistance gene for tetracycline, and then we try to grow that organism on tetracycline, only the ones that got the plasmid are going to be able to grow, and the ones that don't get the plasmid won't be able to grow. Now, the transmission of plasmids often incurs a process called conjugation. You can have special conjugation plasmids called the F plasmid or F factor that encodes for apillis, right? So we talked about this in like the first week. It allows for attachment between two cells, between a donor and a recipient. The recipient does not have an F plasmid. And once they make that connection, the F plasmid can move across or inside of that F pilus. And once it has done so, the pilus dismantles and the cells separate. Occasionally, when we're transferring plasmids, you can get the formation of HFR strains, or high-frequency recombination strains. And this is when the fertility plasmid merges with the bacterial chromosome. And when this happens, the HFR strain can pass copied segments of their own DNA in addition to the fertility plasmid. So here we have an example of an HFR strain forming a conjugation between a HFR strain and an F minus strain. And here it starts to transmit some of the genome along with the plasmid that's been inserted into the genome. And once it does that, it can, it's going to break off. Oh, wrong direction. So once it does that, it can go through recombination where this section gets inserted into the F minus. And then ultimately the genome has some of the HFR strain's genome in it when they break apart completely. There's a nice SEM um, of conjugation. So you can see this pretty cool picture of this pilus being formed between the two cells. Here's another representation of the HFR plasmid with some explanation of what's going on. So in a normal F pilus, or in a normal like conjugation, we've got our F plasmid, which is going to travel through the pilus, and then you're going to end up with two cells that have an F plasmid. If you have an HFR strain, then the F factor has actually integrated into the chromosome. And so when they start to transfer through the pilus, part of the chromosome is going to transfer over to the new cell. And then if recombination occurs here, then you're going to actually get incorporation into the cell, into the chromosome. Now we have transformation. Transformation occurs when bacteria take up environmental DNA. So there's no pilus involved here. The exogenous DNA is just going to pass through the cell wall through a absorption process. And this is only going to happen in what's called competent cells. Competent cells are cells that have the ability to be transformed. Incompetent cells are cells that do not have the ability to be transformed. And so if you have all these fragments of DNA out in the environment, and then you have your recipient cell, if it picks up some of that DNA, like we see in this second kind of panel here, then it's going to potentially go through the process of recombination, which incorporated this big B into the chromosome. The rest of the DNA that didn't get incorporated will be degraded. The process of transformation was discovered by this guy, Frederick Griffith. He demonstrated this in Streptococcus pneumoniae. And he saw that you could actually give non-pathogenic Streptococcus pneumoniae features of pathogenic Streptococcus pneumoniae. And so this is the famous Griffith's experiment. So S. pneumoniae requires this capsule, right, which is this extracellular polysaccharide coat to be infective. So if S. pneumoniae has the capsule, it kills mice that it infects. However, if S. pneumoniae does not have the capsule, it will not kill the mice. Now, the third scenario is if you kill the S. pneumoniae that have the capsule, they also won't kill the mouse. And so what that means is that it's something active, something about the capsule that allows it to become infective, right, and kill. And what he did that was really cool is he killed S. pneumoniae, the ones that cannot cause infection, and he mixed them with non-encapsulated S. pneumoniae. And those non-encapsulated S. pneumoniae were able to start developing capsules. And so that meant that there was something being transmitted between the dead cells and the living cells. And that something ends up being these genes, specifically these capsule genes. And that can happen because the S. pneumoniae is competent It also can happen because DNA is pretty stable and doesn't break down in the environment very easily. And so naturally, microbes are coming into contact with extracellular DNA all the time. They can kind of test it out, pull it into the cell, see if it's useful or not. So here's an example of those four scenarios with the S. pneumoniae again. And keeping in mind that the really important step, I mean, all of them are important because they're controls. Uh, but the part that shows that transformation is happening is the fact that the dead cells that could not cause infection are able to transfer traits to the living cells that couldn't cause infection, making them able to cause infection. Next, we have transduction. This occurs when a virus introduces new genetic material into a host. So they can do this in bacteria. It can also happen in humans and animals and plants. And but we'll talk about bacteria here. The phage can basically add its DNA directly to the host, and it can either result in the formation of new phage particles, or like if we were doing a lytic cycle, or it can go into the genome and you can get a prophage. And in these prophage situations, that's where we get this process of transduction. So here's a general example of what happens. The bacteriophage inserts its DNA. You've got your bacterial chromosome. And if the phage begins to replicate immediately, you just get lysis. But if it inserts into the genome, then when they start to make new phages, they can carry along new host DNA, right? And so that way, when this, like for example, this guy over here has some host DNA in him, and when he infects this new cell, he can inject that host DNA, which can then be incorporated into this chromosome. Specialized transduction occurs when temporary phages introduce their DNA into the host genome. The phage reactivates, and when it does, it excises some of the host genome, and they build that into the phage particles. And so when that happens, you can package some host DNA, and that's what we have here. So in this example, it's kind of a more elaborate example of specialized transduction. So you have your temperate phage going into the prophage stage, and then when it cuts out the genome, you've got a little bit of phage genome and a little bit of host genome. And so all of your phages end up with a little mix. When they infect a new cell, they can introduce that previous host genome into the new cell. So this is something like, could be one of the drivers of something like a phage conversion. So if a bacteriophage were to cut out, say, a toxin from one organism and infect a different organism, they can transfer the toxin over. Now, our last type of horizontal gene transfer we're going to talk about is transposons or jumping genes. They come in retrotransposon and DNA transposon form. They're both found in prokaryotes, but eukaryotes more commonly have retrotransposons, and DNA transposons are more common in prokaryotes. I'd be remiss if I didn't mention Barbara McClintock. She was a famous geneticist that found these jumping genes when studying maize. So That's what actually can give this like natural maize these different colors in their kernels. And so in retrotransposons, there's going to be a reverse transcriptase that makes cDNA from like a viral RNA genome, and then that cDNA gets inserted into the host cell. So that's what we see here. So first we get the formation of an RNA from um, our virus, and then reverse transcription is going to make your cDNA complement to that RNA, and then this cDNA gets inserted into the genome somewhere else. All right. Now these are thought to be viral in origin, but um, in the once they are in the cell, then they can be copied multiple times through this process, as long as there's a reverse transcriptase available to make that cDNA. Now, if you get a transposon that gets inserted into a gene, then it can actually block that gene's function, right? So this would be kind of like a mutation. You'd have a huge insertion in here, right? And so unless this insertion can get cut out, this gene can no longer function. In a standard DNA transposon, they operate in a copy and paste or cut and paste strategy. And so in the copy and paste strategy, you get your transposon, it copies itself and inserts itself into a new place, again, potentially disrupting that gene function. In a cut and paste strategy, it cuts itself out, so it gets excised, and then it gets inserted into a new location. And if it's inserted into a gene, then it can prevent the function. So with that, we should be able to discuss the differences between horizontal and vertical gene transfer, explain plasmids, conjugation, different features of the cell that allow for conjugation, talk about recombination in HFR strains, the Griffith experiment, transformation, transduction, as well as transposons. I hope you enjoyed, and I'll talk to you soon.